PCR is one of the techniques which is used for the purpose of cloning. So that technique is probably the most important technique or most important technique or discovery from the uh, last century, from the 20th century, discovered and reported in 1988. And the guy, Gary Mullis, actually got Nobel Prize for that discovery back in 1993 for medicine. This technique is the most abundantly used technique in molecular biology today. So what is done in this case is a very specific sequence of DNA is amplified. This technique is a modification of DNA replication also. One technique that we just talked about, DNA sequencing, was a modification of DNA replication technique. PCR is another technique just like that, which is designed for replication of DNA. In this case here, the slight difference is the development of these synthetic primers. So one of the kind of a drawback of a PCR is that you need to know the sequence. You need to know the sequence that you're trying to amplify, that you're trying to clone. And the way it is done is by designing small fragments of a single-stranded DNA called primers. So primer is naturally used in the DNA replication. So if you don't remember DNA replication, so DNA replication is done. This is a single-stranded DNA. So double-stranded DNA becomes single-stranded, and then it is added with a small fragment of single-stranded DNA, which is called primer. So that's the kind of a precondition that is needed for DNA replication, the addition of a primer sequence, which is complementary sequence to one end of the DNA. And then the rest of the things are done with the help of DNA polymerase. DNA polymerase recognizes this template DNA and then adds one nucleotide based on what's present on the template. So in case of PCR, synthetic primers like these primers have to be developed in order for the PCR to be successful. So what's really needed in DNA replication, as I mentioned before, it is template, which is the single-stranded sequence of DNA, all the nucleotides, A's and C's and G's and T's. Then it also needs a primer. It also needs some chemicals or buffers. And one very important thing is DNA polymerase. DNA polymerase is an enzyme naturally used for duplication of DNA. And in case of PCR, that polymerase is called Taq polymerase. Why it's called Taq polymerase? Because it is isolated from a bacterium that lives in hot springs at very high temperature. And that bacterium is called as Thermus aquaticus. So it is a heat stable enzyme that functions at very high temperatures, like can work above 90 degrees Celsius. So 94, 95 is not a problem at all for this enzyme. It is that much heat stable. So almost boiling temperature is withstood by this enzyme. Okay. So here's the technique itself. The technique is, this is double-stranded DNA to start with. This is denatured, meaning it is turned into single-stranded templates by heating that DNA in the tube. The two single-stranded DNA fragments are produced as a template. Then temperature is lowered. In PCR, the first temperature is this temperature at 95 degrees Celsius to denature DNA. And that step is called denaturation. The second step is called annealing. So where the temperature is brought down somewhere between 65 to 55 degrees Celsius, that is called annealing, in which the primers that are present in the same reaction, they find their complementary sequences. For example, primer 1 is sitting here and primer 2 is sitting here based on their complementarity. And the third step is called elongation. So annealing is binding of the primer to the template. Elongation, which is the third stage of PCR, it is done at 72 degrees Celsius, and that is elongation or extension. So in that stage, DNA kind of fills the gap which is present right after where the primers are bound. So it will duplicate that part, and this will duplicate this part. At the end, you will get actually those two double-stranded DNA. So this one is this, and this one is this. So it was single-stranded. Now it has become double-stranded because of the elongation stage of PCR. So these are the three steps of a PCR cycle. And it is repeated many times. It is repeated somewhere between 30 to 45 or even more times. The same steps, denaturation, annealing, and elongation, because each stage will produce twice the number of copies compared with the previous one. That is called exponential increase. So 2 will become 4, 8, 16, 32, 64, 128, 356, so on and so forth. So each step will be twice the number of copies compared with the previous one. So after about 25 cycles or so, the number of copies that are produced from a single double-stranded DNA are probably more than 1 million. So just imagine if you're starting with one copy, after 25 cycles or so, from that one copy, you got about 1 million or more than 1 million copies of DNA. This technique is so powerful, it is used for crime scene analysis, where they find a follicle or maybe some skin cells under the nails, or maybe some amount of semen or something on the crime scene, and that is enough to make a big amount of DNA from it. So and can be compared. That can be used to compare with the DNA in the databases. A lot of people have been actually caught and convicted based on this PCR technique, and a lot of people have been exonerated based on the DNA analysis also. Those people were captured decades ago. At that time, more sensitive techniques were not available, but they did actually collect some DNA from the crime scene, and now they analyzed that DNA and found out that their DNA sequences did not match with the suspects. So people who spend there are many cases where the people spend about 3 decades in the jail, got finally released after that many years based on this technique.
Professor, I have a question about if we could place the DNA that we just extracted here and multiplied, can you place that in a bacterium if you wanted to make select proteins, or that's not how that works?
Sure, it does. It does. Okay. Actually, it does. That's how they are cloning nowadays. So the cloning with the help of restriction enzymes is very limited now, because the entire genome has been sequenced. So if you are interested in any genes from the humans, the sequence is known. What they do is they make those 2 primers. They design those 2 primers and just amplify DNA instead of going to restriction enzymes and trying to find them on the plates. They just PCR it. And once they PCR it, they can clone that directly into bacterium. And that way they can find out what type of proteins are produced from them.
Okay, thank you.
Mm-hmm.